Okay, yeah, thank you. So I will be introducing κ-velo, as mentioned, a method to improve single-cell RNA velocity estimation. And my name is Valérie Marot-Lassauzaie. I'm a PhD student in Valérie Marot-Lassauzaie's lab in Berlin at the Berlin Institute of Medical Systems Biology. So maybe first, what is RNA velocity? In single-cell experiments, because of the destructive nature of the technology, we only get a single snapshot of every cell, and we can't actually follow the cells through time. So we can't get multiple measurements for a cell through time. But we are still interested in studying dynamical processes where the cells are actually undergoing changes over time. So here's an example of a developing mouse pancreas, where we have the ductal cells in the beginning that are slowly differentiating to these 4 phases of alpha, beta, delta, and epsilon cells. And even if we did not have any prior knowledge on this system, we would still be interested in finding out which way the cells are moving in this system. And this is where RNA velocity comes in. So it's a concept that was first introduced in 2018 by Valérie Marot et al. And it's based on the fact that in eukaryotic organisms, we can distinguish new RNA from old RNA by the presence of introns. So if we were to follow a single RNA molecule through time, at first it's transcribed with the rate alpha, giving rise to this unspliced RNA. This unspliced RNA is spliced with the rate beta, giving rise to the spliced RNA, who is then degraded with the rate gamma. And then on the second plot, if we were to follow, so we can plot these spliced by unspliced abundances, and if we were to follow one cell through time, we would see that when the gene is turned on, first these unspliced counts go up, and so these spliced counts follow, until we reach a first activated steady state in the upper right corner, where what goes in and what goes out is at equilibrium. And then when the gene is turned off again, first the unspliced counts go down, followed by the spliced counts, until we reach a second equilibrium, where everything is 0. And the RNA velocity is then defined as the time derivative of the splice counts. And this allows us to see the direction of change of these cells, and then also predict the future state that the cells will have. And so a typical RNA analysis workflow, we start with these matrices of spliced by unspliced counts. So we have a measurement of spliced abundances for every cell, for every gene. And then for the same cell, for the same gene, we also have the unspliced abundances. Yes. And then if for every gene, so for one gene at a time, we can plot these spliced by unspliced abundances, and from those recover the alpha, beta, and gamma parameters. And once we recover this for every gene, we can do this for every gene, for every cell. We now get a high-dimensional velocity vector, where we have a velocity value for every gene, for every cell. And because we can't really understand this high-dimensional velocity vector, typically we want to then visualize the velocities on a low-dimensional level. And this concept was first introduced in 2018 by Valérie Marot et al., as I just said. It was then extended in 2020 by Bergen et al. With a new package and tool, k-velo. And we found that this package still has a lot of conceptual and practical issues that make it not really usable and not very robust on different types of data sets, which we describe and revise in our new work. And these issues are on processing, parameter recovery, on the recovery of the high-dimensional velocity vector, as well as in visualization. So during the course of this work, we also compare our results, our new results, to the current state of the art that is still kc-velo. Right, so I'm going to now explain one of the issues to give you a bit of an idea of how we address these. And that first issue is related to scale invariance. So what we do is we solve for these 3 parameters, alpha, beta, gamma, given 2 sets of observations. We have 3 variables that we try to find, 2 sets of observation, meaning we can only solve them up to a constant. In other words, what we have is alpha, beta, gamma is a solution, and this solution perfectly describes this purple almond-shaped dynamics that you see on the plot. But alpha kappa, beta kappa, and gamma kappa describe the exact same shape that we see for any possible kappa. And this is not too much of an issue if we're only looking at a single gene, but once we start to compare multiple genes, this is more problematic. So let's take the example of one cell of which we try to predict the future state, and 2 genes. If we predict gene A to have twice as many counts in the next time step, and gene B to also have twice as many counts in the next time step, but actually gene B should have 10 times as many counts in the next time step, the future state that we predict is not a state that actually exists in gene space. This is not a real cell that can exist. So we predict a direction of change that doesn't correspond to the true direction of change that we would see. So this is now explaining the issue. What can we do? Let's say that we have the recovered parameters alpha, beta, gamma, and the true parameters are now kappa alpha, kappa beta, and kappa gamma. And that we have 2 cells. So let's take 2 cells, UI and UJ, are the measurements of unspliced counts for these cells. And let's assume that we also have delta t, a measure of time between these 2 cells. Now what we can do is we can rearrange the equation of u as a function of t that we simply get from the ODEs, so that we have these equation that you see in red, where on the left we have kappa delta t, and on the right we have only parameters that we know or that we recovered. So in other words, what this means is that if we have delta t, we can actually solve for this gene-specific kappa. Now in practice, we don't actually have delta t. If we had delta t, we would not need to do RNA velocity analysis. So we need to use a proxy of delta t to recover the scaling parameters kappa. And what we suggest to use is the density of cells, or the number of cells between 2 states, as a proxy of delta t. If we have cells that are continuously flowing through a differentiation process or any dynamical process, and we randomly sample 2 cells in this process, then the number of cells between these 2 states is actually a proxy of the time that has passed since the first and the last, or like the second cell entered this process. And what you can see here as well with this example of 2 genes is that this displacement in u is also proportional to the speed of the gene. So we can use this to recover the scaling factors kappa. And this is what we do in our new method. So we validated this on simulation. We simulated a set of genes with varying parameters of reaction rate, and also these genes are then scaled by a true kappa between 1 and 15. And we can see that we do a reasonably good job at recovering this kappa. And then once we have these, we can then look at the high-dimensional velocity vector. And here we're just looking at the example of 2 genes, a fast gene with triangles in slightly lighter color, and a slow gene, circles in slightly darker, once for S in blue and once for kc-velo in red. And what we can see is that kc-velo recovers, so if we only look at 1 gene, kc-velo recovers the velocities very well. These are highly correlated to truth. But once we start to look at multiple genes, we see this pattern that looks a little bit like an over-dispersion. And this is actually proportional to the scaling factor kappa. And in comparison to that, we do a much, much better job of recovering the velocities. Now, bringing this together in the example of the developing mouse pancreas that we saw just before, we can also recover the information based on the scaling factor of how fast and how slow genes change over time. So here we have some examples of fast genes and slow genes. And we can see that fast genes are only upregulated in the end of the differentiation trajectory, or during parts of the differentiation trajectory, they go up and down again. We also have this example of ADK, which is a cell cycling gene that is only active in the ductal population, which we know is much, much faster in this system. The cell cycle is much faster than the entire differentiation trajectory. In comparison, slow genes are slowly up or down regulated over the entire trajectory. Now, once we've recovered this high-dimensional velocity vector, typically we want to visualize the velocities on a low-dimensional embedding. And there are multiple things that we want to represent in this low-dimensional embedding. We want to keep the information of the direction of the vector. That's the most basic thing, which way are the cells moving in this process. But we're also interested in keeping the magnitude of the velocity vectors. So if if cells are moving faster or slower in different regions of the trajectory. And then lastly, we're also interested in looking at local variation. We're analyzing individual cells because we also want to see how do these individual cells change, and we don't want to only look at average trends because it also represents, for example, fluctuations of the dynamics or also cell plasticity. And without wanting to go into too much detail, we suggested that the Nystrom projection method, this is a method that already exists to project new states onto existing low-dimensional embedding, actually much better preserves all of these aspects into to integrate the velocities onto nonlinear embedding. And now bringing all of these aspects together, again in this example of the developing mouse pancreas, we really nicely see this, the velocities, and in particular what I find very, in particular we see the population of cycling cells in the beginning. There's much higher variation in the errors that we see here. And then when the cells are committing to their fate, there are much higher agreement in the direction of the velocities. And finally, we see the commitment to the four fates of alpha, beta, delta, and epsilon cells. Now I want to make a talk about another issue that current RNA velocity analysis workflow has that is related to processing. And I think this is nicely exemplified in this data set of hematopoiesis. So people have struggled a lot in doing RNA velocity analysis in blood. And this has been attributed in large part to genes with multiple kinetics. So what was studied in this data set is we have this population of blood progenitor that differentiates in the erythroid branch. And what we see is that the recovered velocities is a complete inversion from what we actually expect to see from prior knowledge in this population. And then they studied the different genes that were responsible for this, and they found that a lot of genes seem to have multiple kinetics. So instead of having just alpha, beta, gamma, they saw genes that seemed to have a second transcription rate alpha. But in fact, we could show in this work that this is actually an artifact of normalization. What was being done here is the two matrices were normalized separately instead of being normalized jointly. And they should be normalized jointly because the cell size is defined as the sum of the spliced and unspliced count. And this brought in artifacts that made these genes seem like they had multiple kinetics. And when we resolve this, we don't find, for most of these genes, we don't find that they have multiple kinetics anymore. And in fact, on the right, you can see we keep everything of the kc-velo processing the same, but we only change this normalization step, and most of this false dedifferentiation is resolved. So lastly, I would like to bring all of this together on the example of a data set of hematopoiesis. So this is a work that we did in collaboration with Yasmin from the ESSES lab, and this is a data set of hematopoietic stem and progenitor cells. At the top of this UMAP, we have the hematopoietic stem cell, so the most stem-like state that differentiates into these four branches shown in red, blue, yellow, and purple. Green, yellow, and purple. And just to accentuate the direction that we expect differentiation to go, here is from top to bottom. So we have the blue cells as the most stem-like state. Yeah, and so I think it's a really nice example to show how bringing all of these steps together allows us to nicely recover this differentiation trajectory in the hematopoietic system. So we have the most stem-like states in the beginning that differentiate into these four final states shown at the bottom of this UMAP. And in comparison, kc-velo recovers a complete reversal of the velocity directions. So to summarize, kp-velo is able to recover the relative speed of genes. I also introduced a new visualization method to visualize the high-dimensional velocity vector on low-dimensional embedding. And then lastly, I also wanted to point out the importance of careful processing to prevent the introduction of artifacts, in particular in the case of this hematopoiesis example. If you're interested in finding out more, our preprint is out on bioRxiv. The code for this study is also available on GitHub, and we have a small Python package that will be made available next week. I did not completely finish before coming here. And then lastly, I would like to thank my group, in particular Lali and Brigitte, who worked with me to get on this project, as well as all of you for your attention.
Thank you very much for the very nice talk. So you have shown that there are some genes that go faster, some others that go slower. So I have two questions about that. So one is, at the end, you are given a velocity per cell. So how you go from individual velocities of genes to cell velocity? And the second is, do you have some kind of insights of the different functions, some pathways that these different genes represent?
So, yeah, I think there's two things. So to visualize these velocities that we get per gene, there's different visualization methods that we can use. We can visualize the methods on PCA, and in this case, the dimensions are just a linear function of the genes. So this is quite simple. But if we want to look, so this is maybe this kind of representation. If we want to look at this kind of representation, we don't actually have this is not a linear transformation of gene space. So to get this velocity per cell on this type of embedding, we need to find other ways of projecting the cells. And what we can do is, so I didn't want to go into too much detail, but yeah. So this embedding, we recover it in a way that we get the transition probability between all the cells. And this is how typically these embeddings are calculated. And then for the future state, we can also get the transition probability for all the cells, for the cell's predicted future state, and try to then map them onto this existing UMAP. And then one minus the other is then the velocity onto this UMAP. And your second question, can you repeat?
It was about the characteristics of the biological readout of the different velocities per gene. So if there are some pathways that go faster, like you have been trying to characterize which are the fast and slow genes.
Yeah, that's a very good question. I think it's very dependent on the type of data set that you're analyzing. And I think this is why it's very important to really look at which genes are fast, which genes are slow, which genes can at all be used for velocity recovery, because a lot of genes don't have enough spliced or unspliced counts at all. In the example of the pancreas, I think some of the fast genes that we see are actually genes that define these populations that we see in the end. So we have the GCG gene, which is the glucagon gene, or we also had the insulin gene, although it's not in the top 5.
So the fast evolving genes are those that determine the cell states. That's what you're saying. It makes sense.
Yeah, I think so. In this example, at least, because they're only in part of the trajectory. So they are only at the end or in the beginning, and they more, in this case at least, define the cell states more. But I think it must be very dependent on the type of data that you're looking at.
Thank you.
Hi, just a quick question, if that's all right. kc-velo has a lot of functionality for things downstream of the just stream plots, like pseudo-time inference and picking up velocity-based genes. Does this have any implications on some of that downstream analysis?
Yes, that's a very good question as well. So the thing is recovery of the pseudo-time for kc-velo, there's a lot of issues with that as well. So the way that this is done is for every gene, you get a time of the assignment to the cell along this almond-shaped plot. And then these are brought together for all the different genes. But this is probably very likely not correct. So if you have a gene that is turned on very fast and then spends a very long time in steady state, or more importantly, in these cases of genes that are turned on only at the very end, then we know that these are later time points and they should not be assigned early time points. And I don't think that summing up all of these errors in a voting system as kc-velo does it, I don't think they cancel out. So at the moment, we don't have an alternative for that. There's downstream analysis. So CellRank, for example, has nice new approaches on how to deal with this. And they are based on, they use the entire high-dimensional velocity vector to calculate the speed of change in different gene regions. And we think that this is a much better approach to recover time than simply summing together all these estimates per single gene. But at the moment, I don't have a perfect solution yet either.
Thank you.
Thank you very much for a great and very clear talk. couple of very quick questions. For your cell density idea, do you rely on the assumption that you have basically a single trajectory, or it's okay that your cells have multiple trajectories and also actually multiple routes?
Yeah, so that's also a very good question. So in the beginning, we tried this with using diffusion distance, but we had exactly this issue that if there's multiple trajectory, if there's multiple routes, then we really struggle with delineating these trajectories, and also we have this additional source of error. In our current density-based approach, we only look at the density for a single gene. So we don't get this density estimation on any combination of the genes. We're only looking at this US plot for one gene. So we don't have any issues with lineage or multiple routes.
But in 2 dimensions, right?
Yeah, exactly.
Okay.
What could be an issue is if you have a very strong enrichment of specific subpopulation, then this density assumption is violated. Or if you have very strong cell birth or cell death, and at the moment we're working on trying to incorporate this in the model.
My other question is, you spoke a little bit about normalization, and I think that the default for a cvelo, and maybe for the cycle package as well, is to also average between neighbors of your embedding. And I know we tried without it, which would be ideal, of course, but we don't see anything, any meaningful results. Can you comment on that? Can your approach go towards not relying on the KNN graph, at least at this early stage of the process? Because there is a little circle.
At the moment, we still use these average, but we're also looking into getting these input U and S count matrices in a more reliable way, because we believe that there's a lot of issues in the way that these are generated as well. But at the moment, yes, we also rely on this average of neighborhoods in gene space. So that's, I agree, a very strong limitation as well.
Thank you.
Okay, so let's thank our speaker again. You have some questions.
So there are questions online, so we can go over it.
And with that, we close HiTSeq for this year. Well, we look forward to seeing you and your submissions for next year. Thank you.
It's over here if you want to type it or if you want to just like read the questions and answer them. I just don't know if they're still here, but
You might be able to take them with names.
yeah.
How did you
Yeah, the first one I don't know. You can just like type over here and we like find it. Yeah, sure. Like, or it's probably easier if you just like open my directory if they're still here. Oh, it's also up there. The question is also up here. If you go to the right. I have no idea if they're still there.
Yeah, well, I will just answer it. So there was a question in the chat about if I have a sense of whether the method would work for a relatively rare cell type and what the sort of threshold would be. And I think this depends a lot, of course, on your data set. If you have sampled a lot of cells, then it should still work reliably. I think, yeah, what you need to look into is these plots of U by S eminences, and if your rare cell types have genes for which you recover the velocities quite well, and you can see that the density of cells over these US plots is still sufficient, then it should be fine. But